Hi class, this is your instructor, Skylar Huff, and we're here in Chapter 8, How Cells Make ATP, Energy Releasing Pathways. Cells are tiny factories that use the information inherent in their orderly systems to enable them to process materials on the molecular level through thousands of metabolic reactions. Cells exist in a dynamic state and are continuously building up and breaking down many different cell constituents. As we saw in our last chapter, being Chapter 7, metabolism has two complementary components, catabolism and anabolism. Please keep in mind that catabolism is what releases energy by splitting complex molecules into their smaller components or component parts, whereas anabolism is that synthesis of complex molecules from simpler building blocks. Anabolic reactions produce proteins, nucleic acids, lipids, polysaccharides, and other molecules that help maintain the cell or the organism. Most anabolic reactions are endergonic, and they require ATP, and some other energy source to derive them. So with all of this being stated, what I'm getting at, class, is that every organism must extract energy from food molecules that it either manufactures by photosynthesis or obtains from the environment, as you see here. Amazing how that bear, class, is there with his mouth, or well, I'll say his or her mouth open, waiting their, class, for that fish. Let me pause. So animals such as birds and the dragonfly, as you all see here on page 167, obtain organic molecules from their diets. How do they obtain energy from these organic molecules? What an amazing question that may be. First, the few molecules are broken down by digestion, and I would say mastication even comes before, or that mechanical digestion, and then of course comes chemical digestion, where of course digestion into simpler components that are absorbed into the blood by way of the small intestine, to be more specifically, and then transported to all of the cells. So it's the catabolic processes that convert the energy in the chemical bonds of nutrients into the chemical energy stored in ATP, otherwise known as adenosine triphosphate. So it's then subsequently occurring inside our cells, and it's usually through a process known, class, as cellular respiration. So as I say all of this, just keep in mind that it's by the process of cellular respiration, be it aerobic, which of course makes much more ATP, or be it by way of that anaerobic process. Aerobic respiration, I repeat, aerobic respiration requires oxygen, whereas anaerobic respiration, of course, in that vein, class, it of course does not involve oxygen as that requirement. So with anaerobic respiration, included with that is fermentation, requiring no oxygen at all. So here, class, meaning via the process of organismal respiration, meaning by way of our lungs, that provide that steady supply of oxygen that enable your cells to capture that energy through aerobic respiration, which is by far the most common pathway and the main subject, class, here in Chapter 8. So here in this chapter, we'll go through three processes. So we'll go through, class, the three pathways known as aerobic respiration, anaerobic respiration, and fermentation, all of which, class, are exergonic reactions, meaning all are, of course, going to be exergonic, and they release free energy that can indeed be captured, class, by the cell. So as you see here, these are the three pathways, first of which being aerobic respiration, secondly, class, anaerobic respiration, and thirdly, class, fermentation. So this is where we're going. Let us now begin. Redox reactions. So here we have it, class, that cells use aerobic respiration to obtain energy from glucose. You can find me, class, here on page 168. So as this occurs, I'll just put it as being most eukaryotes and prokaryotes carry out the process of aerobic respiration. And this is a form of cellular respiration requiring molecular oxygen class. That is oxygen as being, of course, O2. So having stated that, what I'm getting to here, class, is that during aerobic respiration, nutrients are catabolized to, I repeat, nutrients during aerobic respiration are catabolized to carbon dioxide, I'm changing my color here, to carbon dioxide as well as water. So as I say this, it's that most cells use aerobic respiration to obtain energy from glucose. And I'm making sure to annotate this to make sure you can see what I'm getting at. So here it is, C6H12O6 class, that is glucose indeed. So glucose enters our cells class through a specific transport protein in the plasma membrane. If you go back to your 5th chapter, in chapter 5 class, we discussed how that process occurs. And looking closely, class, we can now see the entire process as it happens, meaning the overall reaction. So given that overall reaction, class, let's look closely at how it happens. So with what's being given, class, I'm going to give you this overall reaction, and I would say, for purposes of our discussion, we'll use this equation, class, in its simplified form to indicate that net yield of water. So let us begin. On the test, class, look for this, and if, in fact, you have any questions, please contact me. So let's first things first, get to these redox reactions, class. The reduction and oxidation, they always occur together. So if you can go back with me and think about the two processes, class, reduction, of course, is to gain. Oxidize, class, is to use. So glucose, class, here is no longer going to be glucose. It will then, class, be what is called CO2. So whereas glucose is oxidized to CO2, I'm just changing my color here. I'll state that, I'll go with that color, something that pops, that oxygen, class, will be reduced. So there you have it. So seeing both of those components, let's now get to this equation, class, a bit more formally. So let's begin. So I'll say C6H12O6 plus 6O2. And from that, we then get 6CO2 plus 6H2O plus energy. And of course, this is energy in the form of ATP. So given what I've just provided you, class, I would say most definitely look for this on your test. You should be able to write out this equation and, of course, identify both the reactants, class, and the products. You should be able to identify, class, I say both the reactants and the products of this being the process of respiration, class. So again, glucose is oxidized, and of course, oxygen is reduced, as shown above, class, in red and blue, respectively. So what is being happening there, or at least what has happened, is that those electrons are transferred to oxygen, class, in that series of steps, class, that we're getting to just right now. So just keep in mind that aerobic respiration is where the electrons, class, where these electrons associated with the hydrogen atoms in glucose are transferred to oxygen. So by way of this process, the free energy of those electrons is coupled to the synthesis, class, of ATP. So let's continue on now, class. And if you have questions about any of this, I say again, you should contact me to let me know what those questions are so I can make sure that you understand what's going on here now. So just keep in mind, class, that via this process of cellular respiration, yes, oxygen is required, and the nutrients are catabolized to form CO2 and water, and most use this class to obtain energy in the form of ATP from glucose. So the changes in the energy class are as follows. It's the energy from glucose molecules, meaning it's kind of analogous to liberating the energy class from a fallen object. And this certainly reminds me of what I call physics class. I don't know if y'all have taken a physics class before, but this reminds me of physics indeed. So it's so much as taking this fallen object here and just using that energy from this, or that total energy class E, comes from, of course, being the same, whether, of course, it occurs from, I guess you say, point A to, I guess you say, B here, or from class point A to point B here. It is all the same, whether it occurs class A all at once, or B class in a series of steps. So let's continue on now, class, with the process. If you have any questions to state, class, please let me know. So what we're seeing here, class, in figure 8.2, shown on page 169, is the entire process as it'll be taught to you beginning now. So with what you see, I'm going to just change my color up to a color that may, I guess, be easily seen. I'll leave it at that. So what's happening is that we have the first part of the process known as glycolysis, number 1. So what happens is we take what is known as glucose, and glucose is split, as the term says, glycolysis. So by way of glycolysis, glucose is split, and it'll be split, class, into those pyruvates, those 2 pyruvates. Yes, there's a bit of energy that's created, 2, of course, molecules of ATP are created, and that's the net piece, meaning it's not, of course, just 2. It's actually a 4, but of course you must invest energy, class, to get energy, just as in money. From there, class, to the second of 4 steps, being, of course, the formation of acetyl-CoA, and to get acetyl coenzyme A is going to happen, class, by way of that link reaction found here. And then the third class of 4 portions will be that citric acid cycle, and we'll do more of that in just a moment. And then we ultimately, class, end up with the last class, the last of 4, with the electron transport chain, class. And please don't forget chemiosmosis, where we, class, get the ATP you see there. So that is the process, class. And to help you with this, there's table 8.1. Table 8.1, class, on page, as I just mentioned, 169. You can use that table to provide a summary of the events occurring. And I will go through those things, class, more or less, with not so much what we start with, but definitely, class, what the products are of each of these 4 pieces. So on your test, prepare yourself to be able to describe this process. Actually, again, on your lecture test, please prepare yourselves to be able to describe the process of respiration as you, of course, are about to learn it. Let us begin. So we have the dehydrogenations and, of course, the decarboxylations and the preparation reactions. As I say all of this, class, we're getting ourselves prepared for the process to occur. So as we remove those hydrogen atoms, they're being removed so they can get to, class, our electron carriers. They're being removed, class, so they can get to these carriers of electrons in a D plus and FAD. And I won't mention much about the decarboxylations. However, these are just those reactions, class, in which the carboxyl groups will be removed. And they're removed from the substrate as a molecule of CO2, which we'll get to, class, here shortly. But it will be by way of that CO2 you see. So let's continue now, class, and get to the process. Bingo. The first of 4, glycolysis. So what happens here, class, is we take glucose. I repeat, we use glucose. I'm at the top of the screen. So just as the name states, it's going to be the splitting of glucose. So we'll take this 6-carbon, I repeat, we'll take this 6-carbon molecule, and it will be split, class, into 2, TWO, into 2 3-carbon molecules of pyruvate. So that's pretty much it. So I say again, we begin with glucose, class, and this, don't forget where it occurs. This occurs, class, in the cytosol, or the cytoplasm of the cell. So in the cytosol of the cell, glucose, class, which is there, will be split, class, from a 6-carbon molecule into 2 3-carbon pyruvates. So just to make sure I reiterate, class, as it states here on the screen, there is no, I repeat, there is no requirement, class, of oxygen. So let's go back to a bit of evolution. Since oxygen is not required, class, evolutionarily, that does mean that this process can occur in what? As I pause, that means this process, class, it can most definitely occur in bacteria, in what I refer to as being those unicellular prokaryotes that have no mitochondria. So keep that in mind evolutionarily. It would be, of course, the process of glycolysis that allow bacteria to, of course, create their energy. In the meantime, class, the process yields 2 ATP and 2 NADH. So, of course, we invest the energy, and, of course, we use, or at least yield, energy. This is glycolysis. So, as it states, class, the energy investment phase, it takes, class, 2 ATP to, of course, split that known as glucose into, of course, those two, I repeat, those two G3P, the glyceraldehyde 3-phosphate. You'll get more to this, class, when we get to photosynthesis. I'll mention G3P again. So the second of which, class, is where we get the, of course, 2 pyruvates and those 2 NADH, which both of which, class, will then leave and move on, class, if, I repeat, they will then leave and move on, class, to the mitochondrion, if, of course, oxygen is there and available. Let's now, class, get to, of course, the second portion, being the conversion, class, of those 2 pyruvates into acetyl-CoA. So I can't stress enough, class, that what just happened by way of glycolysis is we, of course, took that 6-carbon glucose and converted it into those 2 pyruvates. So those hydrogen atoms, class, were transferred to electron carriers. And, of course, we have the 2 NADH. Next up, class, in the mitochondrion. I repeat, next up in the mitochondrion. So I'll write down in the mitochondrion. And of course, I can make it plural, class, and put mitochondria. But since we're now, class, in the mitochondrion, acetyl coenzyme A formation occurs. So the pyruvates combine with coenzyme A. I repeat, the pyruvates, they combine with coenzyme A. As they combine with coenzyme A, class, they form those 2. I repeat, they form those 2 acetyl-CoA. So as they form those 2 acetyl-CoA, class, we also have the release of 2. I repeat, we also have the release of 2 CO2. Lastly, we have 2 NADH. That is the process, class, of the formation of acetyl coenzyme A, or at least 2 acetyl-CoA, by way of attaching to acetyl coenzyme A. So there you see it, class. So this is that known as the CO2 being released. Pyruvates, class, enter in. From those pyruvates enter in, by way of acetyl-CoA, we then, class, have those 2 acetyl-CoA, by way of them combining with coenzyme A. There you have it, class. Continuing now. So up next, class, is the citric acid cycle and Krebs cycle. So we have the citric acid cycle, so tricarboxylic acid cycle, and Krebs cycle. So we're here, class, in the mitochondrial matrix. So still in the mitochondrion, class. I'm just giving you, of course, an idea of where we are in the cell. So I won't go through each of those 8 steps, class. I'm going to give you, of course, an abridged version. So in this abridged version, class, I'm going to give you the products. I repeat, in the abridged version, I will give you the products. So products here, class, are as follows. So we have a total yield. We have a total yield, class. I guess I'll leave it at, it doesn't show them all, so I'll leave it here. The total yield, class, will be, of course, 4 CO2. There's a yield, class, of a total of 4 CO2. Additionally, there will be 6 NADH. I repeat, there will be 6 NADH. There will be 6 NADH. There will be 2 FADH2. Their class will be 2 FADH2. ADH2. And of course, there will be 2 ATP. There will be 2 ATP. So what I'm stating is per glucose molecule that is used, we will have 4 CO2. We will have 6 NADH. We will have 2 FADH. And we will have 2 ATP. So I'm giving you what's yielded, what those products are of the citric acid cycle, or citric acid and Krebs cycle. Now, class, we're here to the electron transport chain, and as it states there, it is coupled, class, to the synthesis of ATP. So as this is happening, we have those electron carriers, NAD plus and FAD. So as you're seeing here, and I'm going to change my color just a bit, they're no longer what I just stated. They're actually now, class, NADH and FADH2. So we have NADH there, and we also now have FADH2. As it states, it's all because of those hydrogen atoms, class, meaning all because of those electron carriers. So what will happen is they will move down the electron transport chain. So as this is happening, NADH and FADH2 will enter the electron transport chain, where they, as I mentioned, class, or those electrons are moving from electron acceptor to electron acceptor, and as they're moving, class, they're moving down to that most electronegative. And what I'm saying is they're moving down, class, toward oxygen. That's where they're going. Oxygen, class, I'll tell you right now, it is that final electron acceptor. So that's where those electrons are moving as they go down the electron transport chain. So with this, some of the energy that is there is going to be used. And that energy, class, that is there being used will be, of course, driving, I repeat, it will be, as it states right here, it will be driving the synthesis, class, of ATP, and that will be all by the process, class, of oxidative phosphorylation. So let's continue on. So in eukaryotes, class, the electron transport chain is that series of electron carriers that are embedded within the mitochondrial membrane. So to say it another way, class, electron transport chains will always be located, will always be within a membrane. And of course, thinking back to prior to now, membranes, class, are made up of phospholipids. So as it happens, class, those electron transport chain meaning components include things such as flavin mononucleotide, ubiquinone, iron sulfate proteins, and cytochromes such as cytochrome C. And let's now continue. So as those electrons pass from electron acceptor to electron acceptor, some of that energy, class, is, I say again, transporting hydrogen ions or transporting protons across the inner mitochondrial membrane, forming, class, a proton gradient. So if you're looking closely here, this lists, class, those 4 large protein complexes that, of course, are there to accept those electrons. So there you have the NADH, there you have the FADH2, and of course, we also have that reduced ubiquinone, and of course, passing them on to cytochrome C, and ultimately, class, you see at that very last bullet, we have, class, the cytochrome C oxidase, which will accept the electrons, class, from cytochrome C and reduces oxygen. I repeat, and reduces what was oxygen, class, to ultimately forming water, class, which is H2O. For instance, I mentioned earlier that the final electron acceptor, class, or I mentioned earlier that the final electron acceptor is oxygen. Well, as those electrons, as those protons, class, pass down 2 by 2 by 2 by 2, they form, class, what is known as H2O. There you have it. So continue on, class. As I stated earlier, oxygen is that final electron acceptor, and a lack of oxygen, class, will block that entire electron transport chain. For instance, it says here, some poisons also inhibit normal activity of cytochromes. One class being cyanide. Cyanide poisoning class can be found out by way of this. Because of course, it would bind to the iron in the cytochrome and it will block the production of ATP. So, getting to the process now to show, give me just a moment here. So it just mentions class, the mitochondria in plants, such as this skunk cabbage. It has that uncouple, meaning it uncouples the electron transport chain from ATP production to generate large amounts of heat. So, in 1961 class, Mitchell proposed that the electron transport chain and ATP synthesis are coupled by that proton gradient that I just mentioned in the inner mitochondrial membrane in eukaryotes. So, of course, what happened is by experimentation, the bacterial cells were placed in an environment with a high hydrogen ion concentration or a high proton concentration, and of course, they synthesized ATP, even if the electron transport class was not taking place. So I won't go over this key experiment class, but this experiment is what showed, of course, chemiosmosis. And I know you're saying, what's this term chemiosmosis? I've heard you say it a few times. What does it mean? Well, it's that coupling of energy. So it's coupling class, the passive transport by way of course, facilitated diffusion, as well as class, that active transport by, of course, I guess I'll just put, active and passive transport with, of course, active transport as well. So you couple both of those to synthesize class, there you have it, ATP. And this was done class back in the 60s. Excuse me. So let's look now at that proton gradient. So as I mentioned class, as those electrons move down the electron transport chain, if you and to look with me class, you can find me on page 178, also looking toward class page 179 with figure 811. Let's begin. So what's happening, class, is as those protons are being, of course, pumped down the electron transport chain, some of that energy class pumps them across, of course, that inner mitochondrial membrane into the intermembrane space. So as it happens, it states low pH. Can anyone state why the pH class is so low in the intermembrane space? So if you go back to pH class, I said pH is nothing more than, of course, the hydrogen ion power, or the hydrogen ion power, or concentration of hydrogen ions, so to speak. So since there are so many more hydrogen ions class in that intermembrane space, that class, of course, is the reason that it is acidic, comparably, of course, to that mitochondrial matrix. So what happens thereafter, class, is, of course, being a higher pH within that mitochondrial matrix, is because, of course, the lower, or at least, I'll say the lower concentration class of hydrogen ions there. So let's get back. So as it states, class, the synthesis of ATP, the protons will diffuse, class. I repeat, they will diffuse from that intermembrane space into the matrix class to the enzyme complex known as ATP synthase. For instance, as the electrons pass from electron acceptor to electron acceptor class, some of that energy is used to transport those hydrogen ions across that inner mitochondrial membrane, forming that proton gradient, which you all can now see there. So as you see that there, it's a lower concentration class of hydrogen ions in, of course, the mitochondrial matrix, and a higher concentration class of hydrogen ions in that intermembrane space. So now we get to chemiosmosis, that energy coupling. It's coupling that active transport as well as that facilitated diffusion to make ATP. So that's what you're about to see. So as it states, class, that central structure of ATP synthase, it rotates, catalyzing, class, that phosphorylation, as it states here, catalyzing that phosphorylation, class, from ADP to ATP, from what was adenosine class diphosphate to adenosine class triphosphate, using that inorganic phosphate. So chemiosmosis allows those exergonic redox reactions to produce ATP class by way of oxidative phosphorylation. So as you see it, class, this complex is turning, taking, class, this hydrogen ion, As it turns, class, it will phosphorylate by that power going from that higher to lower concentration, class, being by way of facilitated diffusion, meaning phosphorylating what you now see, class, as adenosine diphosphate into adenosine triphosphate, creating class 36 to 38 ATP. And that, class, is the process of cellular respiration. And you can see it here in class now. Let's continue, class. So you can see the entire process by way of what energy is produced by way of oxidative phosphorylation, of course, by the oxidation of glucose and getting us, class, to those ATP. So now let's get to, of course, how cells, very briefly, class, regulate aerobic respiration. So glycolysis, class, is controlled by feedback regulation, and it's all based on, class, that enzyme phosphofructokinase. So it has two allosteric sites, meaning it has an allosteric inhibitor site that will bind to ATP when we have very high ATP levels, of course, to stop it. And of course, it has an activator site, class, which of course will use AMP, and that binds, class, when ATP is at low levels. That's enough about that. Let's continue on now, class. We get to, of course, when other energy needs are there. Well, I'm sorry, when other energies are used, or at least other sources of energy are used to synthesize ATP. So other nutrients, class, can be transformed into those metabolic intermediates, the glycolysis, or the citric acid cycle. So I'll begin, class, by changing my color here. Let's go red. So I'll say firstly here with lipids. Lipids, class, can also be converted. And I say for those that would love to be what I call a nurse, a doctor, or just anything in class in a health-related field, please pay especially attention here. What happens, class, that glycerol is converted to a compound that enters, of course, into glycolysis, and the fatty acids, class, are converted by way of beta oxidation. So as I mentioned, class, beta oxidation into acetyl-CoA, class, they will then ultimately enter the citric acid cycle. However, class, this is not the best of cases, because this could, class, lead to the formation, class, of ketones. This could lead to the formation, class, of ketones or ketone bodies. I don't know what happened there. So the formation, class, of ketones or ketone bodies, class, could most definitely change a person's life. And I would say, class, in the worst of ways. Be careful with this, but of course this could happen, class, by way of fasting. This could happen after, I guess, a gastric bypass surgery. This could happen, class, by way of, I guess you could say, being a type 1 diabetic, having not taken your insulin. So please be mindful, class, of, or even a low-carb diet. I would say, or even a low-carb diet. So be mindful, class, of the energy being used to synthesize, class, ATP. And then secondly, class, I'll go to what's up above, and that will be, I'll say second, by way of amino acids. So this will be, of course, by using proteins. And let me go backwards, class, meaning I guess I'll put first here because that's first there. But the ultimate source, class, would be, of course, by using carbohydrates. So I'll put here, class, carbs. Your body would prefers, class, carbs over lipids, and prefers carbs, class, over amino acids, which of course would be by way of proteins. So back to where I was. So first things first, deamination must occur. You say deamination, I did say deamination. So deamination, class, would be, of course, the removal of amine groups, meaning that NH2 from the amino acids. So once that happens, class, the rest of which, class, meaning that carbon chain, can be then used for aerobic respiration. However, class, the process of using this, being amino acids, to synthesize energy, class, will likely not be the case in you or in me, unless, of course, you go through the process of starvation or happen to, class, have been a prisoner of war and you, of course, weren't given food. There you have it. So to, I guess, make this make a bit better sense, what I'll say here is carbohydrates, class, are the first and fastest route. Thereafter, class, you would then use fats, and ultimately, class, proteins by way of amino acids. I say again, the first and fastest, class, would be by way of carbohydrates. This is what the body wants to use first. Thanks a lot, glucose. So this shows, and just keep in mind, I said by using fats, you may very well, class, form ketones. And of course, by production of ketones, you may very well class form ketoacidosis, which is not good. And then, of course, by way of proteins class, I would say that likely will not be used, but the first thing that has to occur class is deamination, removal class of those amine groups. Continuing now we are. Anaerobic respiration class and fermentation. So with this, I'll make it simple. This happens class, as it's stated, anaerobic, it does not use oxygen class. It does not use oxygen class as the final electron acceptor. Typical which class in prokaryotes, this will be environments such as, it says, waterlogged soil, stagnant ponds, or even class in you and in me, our animal intestines. Yes, indeed. Thanks a lot, I guess you say a bit of gas, they say. So what I'm getting at in this vein class is that you should not class have waterlogged soil. That could very well cause your soil to be rendered, I guess I'll say, infertile. Stagnant ponds class should not be stagnant. They should have some degree of oxygen in them. And of course, in animal intestines class, this does occur. So what happens is those electrons, of course, they will pass from glucose. And of course, from glucose will pass from NADH down the electron transport chain. And of course, a couple to form ATP synthesis by chemiosmosis. However, class, it says the inorganic substance such as nitrate, I repeat, such as nitrate or sulfate is going to be class that final electron acceptor. Make a note of those. So oxygen class is not involved as the final electron acceptor, and this is formed class as mentioned by prokaryotes and even class in the intestines of animals. So as we were saying, the end products class of anaerobic respiration are CO2 and one or more reduced inorganic substances and ATP. So let us now continue with where we're going. So here, class, there are only going to be 2 ATP, meaning in fermentation class. I repeat, this is in fermentation. Change your color. So by way of fermentation, what is here is that it's carried out by way of bacteria and some fungi. I'll say it's carried out class bacteria and even some fungi. That's fungi. How that is spelled incorrectly. Spell it correctly, please. And some fungi. So what happens, class, is that there is no, in this case, there is no electron transport chain involved here. There is no electron transport chain involved here. And what happens, class, is there are 2 ATP formed per glucose. So this happens, class, by way of substrate level phosphorylation and by way of glycolysis. So it's inefficient, of course, because it only partially oxidizes what is used, meaning the fuel being used. So what happens is that yeasts are facultative anaerobes. So that just means, class, they can either use alcohol fermentation when deprived of oxygen or, of course, use oxygen. It says facultative either way. So with this, they'll use that acetaldehyde. There's acetaldehyde. And ultimately, class, as they transfer those hydrogen ions to the acetaldehyde, it'll be reduced, class, to ethyl alcohol. So that's why, class, Bush has their own yeast species, because those yeasts carry out alcohol fermentation when oxygen is not available. And of course, this is, of course, the process by way of making A, wine, B, beer, and of course, also this is used, class, in baking, i.e., that's why, class, bread rises. So, of course, certain fungi and bacteria, they perform lactate fermentation. Yes, I did hear that. Lactate fermentation. And I know you all know a good bit about this. And if you don't know what I mean, let's get to it now. So to get to this, what happens, class, is by way of lactate fermentation, well, I'll just put it out there. Lactate fermentation, class. In this, lactate is used, I guess I'll put it as being lactate is used in the formation class or creation of yogurt. And I like a bit of yogurt in the life of me. Especially, well, I won't mention specific brands. So with that, it also had class happens by way of us in our vertebrate muscles, meaning vertebrate muscle cells produce lactate when oxygen class is depleted during exercises, such as you're doing many reps. And I'm particularly thinking class of anaerobic exercise. So when you get to the final, I guess you say final few reps, and then of course you get to the process, or at least not process, to the point at which you cannot go any further. It is likely class because that buildup of lactate within those skeletal muscles. And the very same way, which you can now see class both processes. A, this is ethyl alcohol fermentation, and B, class, this is lactate fermentation. One thing that I failed to mention, class, is that by way of exercise-induced fatigue, as I was just stating, class, and then, of course, getting cramps thereafter. And it's all because of insufficient oxygen, class, and lactate buildup. So I can't stress enough, class, please prepare yourselves well for this exam. This has been Chapter 8 and your instructor, Scholar Huff. If you need any assistance, please reach out. Email me, stop by the office, call me. But above all, class, please study and, of course, prepare yourselves to be able to describe the process, class, of respiration on your test, as well as, of course, briefly, of course, the process of fermentation. Thank you all for listening, and this has been your instructor, Scholar.